At the outset, I want to thank all Russian Ayurveda Congress for inviting me and also for allowing me to present my talk through the video conference. Because of little sickness last week, I was unable to travel and I am missing wonderful climate and wonderful hospitality of this great city, Moscow. I am going to talk about genetic basis to Prakriti and role in personalized and predictive medicine. As we know, we all look different, think different. Human can be classified in different ways, and historically we have been classifying human beings based on physical traits such as shape of the head or color of the skin or hair form, how your body built is there, how is your stature. We can also classify human beings based on anthropological classifications, based on ethnicity, based on racial types like Negroid, Caucasianoids or Mongoloids. These are all different classifications also influenced by geographical, environmental and climatic variations in specific regions which result in genetic and epigenetic changes which lead to different kinds of physical traits. We are aware of relationship between genes and behavior. We know that behaviors are complex traits involving multiple genes, and for any typical physical appearance, disorders or behaviors, genes are not in isolation. The environmental factors are involved in the development of any trait, and this branch of science is known as epigenetics. Actually, when we were working on understanding genetic basis of concept of Prakriti in Ayurveda, epigenetics was not on horizons. So our main work was related to genetic basis. Behavior and personality types. Many of you would know that broadly people can be classified into two different classes, type A and type B personalities. Type A personalities are normally hostile, they are impatient, they are competitive in spirit. Type B are calm, cool and just take it easy guys. The behavioral patterns are important. So these typologies are classified as per different ways. One of the way, which is quite ancient actually, is mentioned in Gita, which is one of the classic from Indian tradition, which classifies human beings as sattvic, rajasic and tamasic. Hippocrates also has classified human beings into 4 different categories like black bile, yellow bile, blood, phlegm. Galen also has classified as mechanocholic, choleric or phlegmatic and sanguine. Traditional Chinese medicine classifies human being into 9 constitutional types. Korean Sasang classifies into 4 types. Thus, there are various ways in which people can be classified. The famous theory of somatotypes, which was proposed by Herbert Sheldon in 1940s, which is based on 3 germ layers of embryonic development called ectoderm, mesoderm and endoderm. People are thin, which are ectomorphs, which are muscular, they are known as mesomorphs, and people who are fat, they are called as endomorphs. Next. While we know all these classifications, still classifying human population for the health or disease purpose is not really very easy, because despite several approaches, there is no reasonably sound way to classify human population. All of us are different, but still genetically 99.9% we all are same. We may differ in physical appearance, we may differ in psychological types, we may differ in somatotypes, we may differ in behavioral types or even anthropological types, but still genetically we are all same. Individuals look different, think different, they have different body structures, metabolism and so on. At the same time, we were still believing that we will respond to a medical treatment in a similar manner. Now, how is it possible? It was not known until a new science emerged called as pharmacogenomics. Now, Prakriti, which is a typical Ayurveda concept, Ayurveda classifies human population into 3 categories which are based on the dosha, and these Prakritis are most comprehensive because they cut across all these different kinds of classifications which we saw. whether anthropological classifications or psychological classifications or ethnic classification, Mongoloids or Negroids or Caucasianoids. So, there are Prakritis, 3 types of Prakritis, for example, in Negroids also, in Caucasianoids also, and thus we feel and we hypothesize that Ayurvedic basis of Prakriti is one of the very good way of classifying human population. There are several ways in which it can be done, and before we go to that, let me first give you a very quick introduction to Ayurveda is. Ayur stands for life and Veda stands for knowledge. So, actually Ayurveda is science of knowledge or science of life. It is in existence for last 4,000 years, and many people believe that it has history even beyond that. The Ayurvedic concept of Prakriti, there is a relationship between the Mahabhutas. There are 5 Mahabhutas, and these Prakritis are formed because of various combinations of these Mahabhutas. Ayurveda classifies human race into 3 doshas called Kapha, Pitta and Vata. Next. Earth and water as Mahabhuta, together they express as Kapha Prakriti. Energy as Mahabhut is represented by Pitta Prakriti, and air and ether together form Vata Prakriti. Now, Prakriti characteristics are also very interesting. Physical characteristics, the lean, medium or well built, the skin structure, whether it is dark, whether it is showing superficial veins, or whether it is showing tendons, bright skin, how it is oily or whitish, how hairs are, whether they are rough, scanty, soft, how their nails are, whether they are dry, thin, small, pink, oily, or psychological characteristics. How is your mental nature? How is it you are intelligent? Whether you are slow, whether you are dull, and also physiological characteristics such as your appetite, your food habits, whether you require small quantity, you eat fast, how are your bowel habits, whether your stools are hard, dry, or how is your digestion rate, whether it is consistent, inconsistent, whether it is high, low, and movement, how you are fast in movement, whether you are moderate or slow. All these things are responsible for Prakriti. So, broadly these are the 3 different dosha Prakriti, and we hypothesize that this dosha Prakriti has genetic connotation. Next slide. If you see Ayurveda therapeutics, very interesting it is. There are various ways in which Ayurveda therapeutics considers variables. Like I told you, there are 5 Mahabhutas, there are 3 doshas. But in addition to them, there are many other variables like dushti. There are 2 types of dushti. There are 7 types of dhatus or tissues. There are 3 different types of malas or excretory products. There are 13 different types of agnis, 13 different types of srotasa, which are microchannels actually. There are more than 700 different dravyas or materials which can be used as medicine are known. There are 20 different pairs of guna, 10 pairs of guna, total 20 gunas. There are 6 rasas, 2 virya, vipaka, then there is prabhav, and desh, dushya, bala, that is the power of the individual, kala, that is the period, time. All these together are considered when we are understanding the nature of Prakriti. It is not easy to find Prakriti of individual, but recently a new software has been developed by University of Pune, where I come from, in collaboration with CDAC, agency which developed India's first supercomputer, and the software is known as AyuSoft. AyuSoft is a novel concept where strengths of Indian traditions are supported by technological advances. Next. As you know that Ayurveda, there are various texts, and these are classic texts. There are 3 classic texts called Charak, Sushruta and Vagbhat, and the 4th classic which talks about diagnostics is called as Madhav Nidana. So, what we have done actually is from these 3 classics and the 4th Madhav Nidana, over 25 or 20,000 different rules are explained in this text. These rules have been converted into software using different kinds of algorithms. And so, AyuSoft actually, one of the component of AyuSoft also can determine your Prakriti. It gives multiple choice questions, and as you go on answering those questions, they have different weightages, and at the end you will be able to know what is your Prakriti, not only your Prakriti, but which kind of food is good for you, in which season what you should eat, what you should avoid, which we call it as Pathya Pathya. All this dietary advice, health advice is also available in Ayusoft for your ready information. Next slide. Next slide. I'm going to skip some of the slides here because the Ayusoft is available on the website, and those who are interested knowing what Ayusoft is can really refer to that. Next slide. So, like for any genomic development, you require a bioinformatics base. We developed first Ayusoft as a software before going to Ayugenomics or understanding genomic basis of Prakriti. Next slide. In pharmacogenomics, I told you that people have identified that we all look different. Even Sir Wilhelm Osler in 1892 said, if there were not great variability among individuals, medicine might as well be science and not an art. And the same slogan was actually expressed in a different way by Alan Roses, who wrote in Nature in 2000 when Human Genome Project was just coming up, that if Wilhelm Osler was alive today, he would have reconsidered his views of medicine as an art and not a science, because we know precisely now genetic basis, and that's how. But still, we may know the genetic basis of a human being, but medicine practice still will continue as an art, but medicine as science has evolved because of these advances in genetics and biotechnology. This new branch called as pharmacogenetics is developed, which really considers that for every individual you may need different therapeutic strategies. One drug may not be suitable for all. Next slide. This was also told by Charak almost 4000 BC, when he said every individual is different from another and hence should be considered as different entity. As many variations are there in the universe, all are seen in human being. So, this Charak quote actually forms basis of pharmacogenomics. Today, we are dealing with syndromes and not just diseases, like cancer, rheumatoid arthritis, asthma, they are polygenic diseases, they are not one target diseases, And therefore, the therapeutic approaches to handle these diseases are bound to be complex and are bound to be different. There are various examples. I am not again going to give examples here, codeine and the drug metabolizing enzymes, because for example, 7% of Caucasians have a variant version of CYP2D6, which is a drug metabolizing enzyme with no activity, and codeine does not work in these people. That means same drug can work very good in some set of people, in some set of people it will just not work. To understand all this is not very easy. Next. We raised a question that is there any genetic basis to Ayurveda Prakriti concept, and are doshas quantifiable? Can we measure doshas? Because according to Charaka, doshas are substances, and if they are substances, we hypothesize that they should be measurable, they should be quantifiable, and they should be considered as qualitative and quantitative traits for the human being. They could be estimated, and to do that, we just did a very small experiment, because our group works in rheumatoid arthritis, and rheumatoid arthritis and HLA relationship is fairly well known. We carried out a very interesting study in which we took 76 patients of rheumatoid arthritis and we took their blood, and we analyzed their blood. We actually analyzed their DNA for HLA DRB typing, which we call it as DRB HLA typing, and we showed on the screen what you see. There are three different types which are clearly emerging out, and they are statistically different. These are Vat, Pitta and Kapha. So, these Vat, Pitta, Kapha allele distribution among Vat, Pitta, Kapha was statistically different. This was for the first time we established scientific basis or scientific support for concept which is emerging from traditional science like Ayurveda. We published these results in Cold Spring Harbor Symposium on Pharmacogenomics in 2004, and this was published in Journal of Alternative and Complementary Medicine in 2005. These papers are available. Those who are interested will be happy to share with them. Next. We also studied Ayurvedic concept of metabolism. For example, Ayurveda says that Pitta Prakriti are fast metabolizers. Kapha Prakriti are slow metabolizers. So, to test this hypothesis, we took CYP2C19 polymorphism as one of the tool, like in previous experiment we had done on HLA. In this case, we did it on drug metabolizing enzymes, and we showed that those alleles, in next slide you will see again the similar kind of a pie chart, wherein the distribution of alleles, those alleles which are very specific for fast metabolizers, were present in Pitta Prakriti. So, this was another example or another support or another proof in support of traditional knowledge like Ayurveda, wherein people can be classified based on their Prakriti. So, Prakriti and genetic relationship was fairly established by our group during this period, and now many studies are coming up to understand genetic basis of Prakriti. Earlier we used to work only on one gene, now people are working on various genes, sets of genes, and also the whole genome studies have been carried out. So, the paper which we published in 2005 work has led in many directions, wherein some of the Indian scientists have been able to publish in prestigious journal like PNAS, wherein they showed that EGLN involved in high attitude adaptation revealed through genetic analysis of extreme constitution types as defined in Ayurveda. Next slide. So, we call it as Ayurgenomics, and this Ayurgenomic project is now undergoing in India at various different centers in Pune, in Manipal, Indian Institute of Science, Bangalore, and CCMB in Hyderabad. Such prestigious centers are involved, and the whole genome studies are being undertaken, gene expression profiling studies are also being undertaken, and results of all these will be available very soon. Next slide. So, the significance or the novel approach to classify human population and its correlation with genotypes and phenotypes, which cuts across racial and ethnic basis, is a unique feature of Ayurvedic concept of Prakriti. Next slide. In nutshell, Prakriti examination actually consists of over 878,000 variables, and such variations to quantify is not an easy task, but Ayurveda has made it easy through their own Prakriti Parikshan, and while the genetic analysis is important and we should continue to do it, the simplicity of classification of human beings based on Ayurveda, or what we call it as Ayurgenomics, we believe that is going to be the key for future personalized medicine. Next slide. So, in conceptual model, there is Ayusoft, a software which can analyze your Prakriti. You have a database which talks about your phenotype, which is a disease Prakriti. There is a database which is called as Ayush or Aushadhi database, which can give you selective herbs which are important to balance the Prakriti vitiation or dosha vitiation, and at the end the prescription will be either drug or diet or lifestyle. This is what actually we call it as personalized medicine. We have done a lot of work on evaluating Ayurvedic drugs in clinical research, and we have published several papers on even randomized clinical control trials. Next slide. But can we really put Ayurveda in capsule? That question we want to raise, because Ayurveda is not just drug. Drug is one component of Ayurvedic treatment, and Ayurveda has, in addition to that, several other like diet, lifestyle modifications, mind-body relationships, all that's put together, and therefore we call Ayurveda as a holistic science. And for studying Ayurveda, a typical randomized controlled clinical trials may not be suitable, and we will have to develop new holistic trial platform to study efficacy of Ayurvedic procedures and Ayurvedic treatments, and not just Ayurvedic drugs. Next slide. Our work has been awarded in 2013 a prestigious Integrative Medicine Award by the European Society for Integrative Medicine, and this work was published in British Journal of Rheumatology. Again, the reprints are available for those who are interested. Next slide. The last part of my talk is about the new age of integrative medicine. Today, medicine wants to become actually predictive, so that we are able to diagnose very, very early. For example, when we are able to diagnose diabetes, already the disease is set. Can we diagnose such diseases much early before they are set? This is called predictive. And preventive, if we are able to really predict diseases early or diagnose diseases early, then we are able to avoid those diseases also, which is called as preventive medicine. And once we have predictive and preventive components in place, then as per the individual requirement, as per the individual Prakriti, we can actually treat every individual, and not all people will be treated by the same drug. This is called person-centric or personalized treatment, which is a typical treatment pattern of Ayurvedic treatment. So, we are saying that there should be a golden triangle, wherein Ayurveda, the strength of biomedicine, including genetics and biotechnology from science, should come together to come out with predictive, preventive and personalized medicine. Many people talk about it as PPP medicine, and there is a lot of good scope to do collaborative research in this area. Next slide. We are doing some work to identify early markers. For example, in Ayurveda there is a concept called Shat Kriya Kal, and they believe that every disease progresses through 6 stages. So, if we are able to identify a person in the first stage going towards disease, then we are able to really prevent further progress of the disease, and this is something which will be very important to work on. Next slide. So, the integrative whole system approach of Ayurveda as expressed by Ayurgenomics and personalized medicine, we believe that it is a key to the future medicine. Next. This poster I want to show before I end my talk. On this poster, we call it a Systems Ayurveda poster, in which there are 230 knowledge points from Ayurveda, and this is just a broad expression of Ayurveda, broad expression. So, 230 knowledge points connected to each other through forward and backward linkages based on their entity relationships, and this poster may remind you of metabolic charts which you see in the laboratory, but this is for the first time we are able to really show the complex relationships between various variables of Ayurveda in systems biology classification way. Next slide. We know that what we are trying to do is extremely difficult, but we get inspiration from people who say that it is only due to internal urge that an achievement can take place. That modern corporate language, they call it as fire in the belly, which was said in Yoga Vasishtha almost 5,000 years ago. We know it is difficult, and therefore, because it is difficult, it has to be done. Next slide. I would end my talk by quoting from Charak Samhita. Ayurveda owes its call not to selfish goals or worldly pleasure, but to compassion for fellow beings. In seeking to know my legacy, you have but seen the leaves of the universal tree, which are too vast for your eyes. May your sight grow and your quest never end. This journal we edit, and those who are interested can visit the website www.jaim.in, and we welcome articles in this journal, and I have already indicated to the organizers that we will be happy to publish proceedings of this conference in this journal. This journal is indexed in PubMed, Scopus and many other important scientific databases. Those who are interested may like to see the next slide, which is a book which will be published just after 2 days on 10th of April by Elsevier, and this book talks about integrative approaches for health. What I have talked here can be found in this book in much more details, and I would request you to visit site of Elsevier and just read some short description of the book, and do acquire this book for your libraries. These are the people with whom I work. What I talked and shared is not my only work, but it's outcome of work from all different types of colleagues coming from different countries and different disciplines. Again, at the end, I would like to thank All Russian Ayurveda Congress for inviting me and giving me this opportunity to share my thoughts and our work on Ayurgenomics and personalized medicine with this distinguished gathering. Thank you very much.